What's up, everybody? Welcome to your Weed Stories on the Craft Growers Network. Today, we are joined by Phil Estrin. He's the tissue culture expert from Kinomic and the Grateful Head. We're excited to learn about his story, and we think we're going to learn at least a little something about tissue culture today. So, welcome to the show, Phil.
Thank you for having me.
Oh, and of course, we've got our Slovenian hobbit, Sasha, joining us.
Hey,
He's
everybody.
our European correspondent, and he's also very interested in learning about some tissue culture. So, Phil, why don't we start by you telling us a little bit about your cannabis story, how your first experiences in cannabis, and how it led you to tissue culture.
Okay, sure. I think my first experiences are pretty common, but just a bunch of guys who are my friends, hanging out, smoking, not knowing if I'm doing it right the first time, coughing more than I expect, and then eventually it clicked. I think by the time I was in my early 20s, cannabis had found a mainstay in my life. And I was fortunate enough to be studying genetics at that time. And there was an opportunity. I'm originally from the state of Connecticut. It had just expanded into medical. And there was an opportunity when I was in grad school to work at a medical facility. And that was really the gateway that showed me that it could be a career path and then more than just a personal lifestyle, something that can be a community and additive. And since then, I've never thought of working in any other industry.
What was your role at that cannabis facility?
Sure. So this was a extraction laboratory. So basically, I was just like a low tech. So Connecticut, very funny. In the beginning, they did not necessarily know how to legalize cannabis and how to legalize these kind of auxiliary things that would be associated with cannabis, like extraction, stuff like that. So initially, they did not allow any organic solvents, including water, essentially. And that's pretty tough in a facility outside of the extraction lab, not being able to use water. So they quickly remediated that. They allowed alcohol, but you couldn't use hydrocarbon. So in Connecticut, at least in 2015, 2016, all they were using was supercritical CO2 because it was solventless at that time and didn't know about necessarily the techniques that we have nowadays. And I was doing that. I was ramping up supercritical systems, running them very far away from my biology background. But they had initially had interest in doing tissue culture. And they asked me to figure out how to do it, essentially, which is obviously what ended up leading me into this specific pursuit.
Nice. And where were you studying? Was that in Connecticut?
Yeah, University of Connecticut.
Nice. And Sasha, you're muted, I believe.
Sorry. Beside the ethanol and CO2 supercritical, did you also use butane extraction? Butanes?
So at that time, you could not use any hydrocarbon. You couldn't use anything, really. Yeah.
What about today? Is it legal to use butane today in Connecticut?
So, Connecticut's in a very interesting state. It's a little bit of, so for reference, in 2015, 2016, when I was there, it was only medical. They only had 4 producers. Since then, they've expanded to adult use, and they've expanded to 4 producers. So it's the same people that were providing for medical have now split their product between medical and adult use. The rules around adult use, I'm not exactly familiar with, but I am pretty confident that medical is still only allowed to be solventless. So now with the live rosins and stuff like that, there has been an expanded portfolio, but there's no closed loop systems or stuff like that.
Okay. And when you were studying, you were studying biology, is that what you said?
Yeah, so I went to school for molecular biology, basically genetics.
And what were you interested in studying before you got into cannabis as far as molecular biology?
Sure. I think in general, genetics had always fascinated me. The ability to kind of meet the moment when chemistry gained some sentience, even though I think There's a lot of very good chemists out there who would say there's even prior examples of that. But for me, that was the first thing that clicked. I had worked in immunology labs before and done a lot of bacterial work, a lot of genetics work, a lot of viral work, stuff like that. All very fascinating. I think, especially when we apply these techniques to medical research and stuff like that, there's a lot of potential for therapeutics that traditional medical approaches couldn't necessarily solve or target. So that was always very fascinating to me. The idea of working for a company like AstraZeneca or Pfizer was a lot less appetizing to me. So ultimately, that cannabis being an alternative pathway was very tantalizing.
And when you were asked to learn about tissue culture, how did you start that journey?
Sure. So I was still at the University of Connecticut. I was in the genetics department, finishing up my master's, and while doing this this work at this medical marijuana facility. So when they asked me, I just went to the plant science department and asked a professor by the name of Gerald Berkowitz, who was doing tissue culture on a plant called Arabidopsis, a model organism for plant science, but not necessarily a commercially or consumer common crop. So I asked him, and he was like, I have no idea how to do it, but why don't you just come and like study here and maybe we can figure it out together? And I was like, why don't I just come and study here and we can figure it out together? And so I very promptly left the opportunity I had at the medical marijuana facility and continued to do research under Gerald Berkowitz's lab at the University of Connecticut. And there, at that time, only the Lada publications from Old Miss were accessible. So that was our starting point, as I believe many people's starting points back at that time. But we were fortunate to find some things that worked. And once my time finished with at UConn, we came and started Kinomic.
And so a question is, was it allowed? And I mean, did that even matter to experiment on cannabis at the university since you did not have a license through the university?
Okay. So, yes, it was allowed because in 2016, they had passed the Farm Bill, which was the first time that they allowed research institutions and agricultural institutions to research hemp. CBD, hemp, like no one was doing the THCA nifty loopholes that you might see nowadays. No one was really talking about D8 and stuff like that. It was just, let's find some fiber seeds. Let's find some like auto-flowering for seed production seeds of hemp. We don't really care. Let's just get some material. Like, I think one thing to note is, many times in biology, it is not a game of what's best, but a game of what's first. E. coli is a classic model organism, not because it is the best bacteria, but because it is very easy to find. It is in all of us, so that makes it very easy. There might be, arguably nowadays, much better model organisms to build from, but E. coli was first. So we were in the same place. We just needed whatever. We didn't, like, we would love to have worked with really good quality substances, but sourcing material was very tough, and it very much mattered. I talked to a lot of lawyers because UConn is Connecticut's flagship public state university. Their legal staff is essentially the assistant AG and stuff like that. All of them wanted to talk to us and make sure that we were interpreting the Farm Bill correctly, obviously not being lawyers and stuff like that. Eventually, long story short, they okayed us to do it, and UConn has a very successful hemp program ever since.
And so, were you actually using like low THC hemp, or were you using?
Yeah. So I mean,
the process is the same.
Yeah, finola. I mean, much to the point of like using today's justifications. Like at that time, we're not flowering anything. You know, tissue culture doesn't require any flowering, so whatever material is tested will show up with the same THC as corn, tomatoes, whatever you want, right? So from that perspective, that's what the Farm Bill kind of limited us to, and we very much played within that bounds.
You're muted.
Can you tell us which strains of hemp were you using? You said Finola.
Yeah, so later on, I mean, again, when we initially got these, they were just whatever was accessible. Eventually, once it came time to publication, a lot of journals won't necessarily accept it's weed, trust me, bro. So it was sent in for genetic fingerprinting, and the closest related other fingerprinted plant was Finola. So it was just subscribed as Finola 2, and we continued onward.
It's a very early flowering hemp variety. We use it here also. We use quite a lot of hemp here, you know, like
Yeah.
Finola is like, you know, it's like a standard here. It's like every third field is Finola here because it's so early, you know. So,
Yeah, it makes total sense. At some point, when we eventually switched to American crops, we had like cherry wine and abacus, I believe, was another one. Yeah, at this point, unfortunately, my memory is not so great. And 2015, 16 is becoming further and further away every year. But I believe those were like the major ones. And yeah, undeniably, the Finola was in many ways very nice because it's fast growing, it's not very hybridized, so it just puts out a lot of plant material, not necessarily the greatest morphology, but a lot of morphology. Cherry wine abacus were they were more realistically just like traditional cannabis strains that had generated a knocked out version of the THC synthase, so now they immediately became hemp, and that made them very commercially viable because they had really good metabolic pathways, they produced a lot of CBD, and they looked like commercially viable indoor plants versus Finola sometimes is not.
And you mentioned that the papers were published. Is this something that anyone can find?
Yeah. I mean, to secure funding, you got to keep publishing in academia. So ever since I've left, they've been publishing like crazy. And if you search Berkowitz, you'll find Berkowitz Cannabis. You'll find a lot of good interviews that he's done, and a lot of his publications.
And so, like you said, they are continuing to study tissue culture and hemp and cannabis.
I think the reality is when you look at something in academia and in publications, it's called an impact factor. You can look at any journal that you want, plus the word impact factor, and it'll tell you a number. If that number is above 3, it means that it's at least peer reviewed. So a lot of times, a lot of very funky science from a lot of obscure papers will get like a 1.2 publication, a 2.3. It means no one looked at it. There was no journalistic insight, right? The impact factor typically means impactful to people. So medical journals are always going to be at the highest of the impact factor because anything they discover in medicine is immediately going to be applicable to many, many people. So New England Journal of Medicine is one of the highest. The Journal of the American Medical Association, JAMA, is the second highest. They're crazy. For comparison, something like Science is a 10. The other ones that I just listed, they're 50 plus. They're radically different ballparks. It's the highest one in plant cell or plant science, depending on which one, or plant physiology, they're at like 4.5, 5. So you can see traditionally plant research is considered pretty low impact. Cannabis has an incredible benefit in being a therapeutic plant as well, so it traditionally scores much higher. So if you are a plant department in desperate need of funding, jumping onto cannabis is an assured way to bump up your publication scores and potentially secure better funding and grants in the future. Connecticut is not an agricultural state, so cannabis being studied at UConn provided the plant science department, which was a relatively minor department, I think a lot of opportunities that traditional research in like aronia, which is a plant I had never heard of before I worked in that department, would probably not bring to them. And that was one that they were studying. And I have not heard of it since.
It's interesting that you said that plants don't get that high of a rating. But does that include food?
Yeah. I mean, like,
because you would think like food is medicine, and you know,
it
next
really
to water, it's one of the most important things we have on this planet.
It is, but you know, at the same time, science is not altruistic. Someone is footing the bill, and this always leads to a large, concerning conversation on ethics, right? In the good old days of Isaac Newton, whatever, all of those people, every scientist we've heard of from before 1800s, came from an incredibly wealthy family and usually had a castle that they were doing all these stuff in their basement of, and that's why they all pursued alchemy. It's obviously been more democratized since then, but it still needs to be funded. And the problem is like corn or agricultural crops in this country. Like corn is the largest one that we plant. 70% of plantable corn is not for food, right? It's for ethanol production, grain feed, stuff that might eventually lead to food being produced, especially for cattle production, but ultimately not calories meant for human consumption. That ethanol producing corn gets a lot more research into it than publicly than food grade corn. And food grade corn is mostly researched by Bayer and large scale ag companies, and their incentive to share that research is limited. So it's a very fascinating, frustrating web. But I think that's like, if you have your own lab, you can study anything you want. I think that's one of the beautiful things about it. There are a lot of, for example, easy to cultivate organisms like fungi. There are a lot of foragers out there who will go with a petri dish and they'll isolate fungal isolates, right? And then from there, if they want to sequence it, that's relatively cost effective. If they want to do that in computational biology, all that's accessible. But to put an academic lab into that, it would be hard to incentivize them unless there is financial incentive, and cannabis offered that to a large amount of people.
I always say that I'm recording this podcast for me, and if everyone else likes it, then good. And I find what you're saying is it's awakening. So, I'm enjoying the conversation.
It's a perspective of somebody who's like a lab rat, right? Yeah. I mean, you're somebody from science. Yeah. So people from science and labs always have a perspective which is more realistic than any others. You know what I mean? It's like you'd say, money rules these kind of markets, right?
Yeah.
It's like that. It's just even the science behind it. It's ruled by big investments, and that's how it's going, right? I mean, the world is going like this today, so we should be aware that it's going on like that, yeah?
Yeah, exactly. Yeah. And I think similarly, like, many good people can be swayed by many directions. Sometimes someone can make a decision that we don't agree with, that we might think is moralistically bankrupt, but they have alternative forces that are pushing them to make these decisions. Family members, financial crisis, whatever it is. We should be as understanding as we are. Ultimately, science isn't good or bad. It's purely discovering facts. A lot of people, like Madame Curie, discovered some of the most important facts in terms of radiation, how it can develop. She ultimately paid the price for it, and her notebooks are continue to be so radioactive that they have to be locked away and can only be shown behind display, right?
Yeah.
That is a very altruistic approach to it, and yet it's still ultimately she sacrificed her life. I think we're very fortunate to be playing with cannabis, and while our discoveries are fascinating, they, stand on the shoulders of the people who came before us. So,
I think you started to talk about before about how you went into the program in Connecticut in the university with this professor. Can you describe your first years? How it was for you? The failures, you have to make a lot of failures, I think, right?
Sure, yeah. So the department that I went to was very, very nice, very accommodating. Everyone there was very curious about us starting to pursue into cannabis. They all wanted to offer help, and me, having never worked with plants prior, was extremely grateful for any help that could be brought. So the way that you tissue culture Arabidopsis is different than how you tissue culture tobacco, different than how you tissue culture tomatoes. And these were plants that people were working with in the department. So they all approached me and were like, maybe this hormone, maybe this concentration of hormones, maybe this media base, maybe that. A lot of failures, a lot of frustrations, especially early on. Very insightful sentiment is, we don't know what we don't know. So I had no idea where to even start. I had no idea what was deficiency, what was toxicity, what played well together, what was synergistic, what was antagonistic. Eventually, though, you get your first success. Classically, I think the way to build it is like science is incremental, success is incremental. You get 5% success before you get 10% success, before you get 20% success, before you get 40% success, before you get 80% success. And then at some point, you're just figuring out what your margin of error is. Is it 99%, 99.9%, 99.999%? And at some point, you consider yourself to have successfully tissue cultured the strain, the plant, however broad your successes are. But I can promise you in the beginning, it was far away from where we are now, almost 10 years later.
Did you
You
know?
said this was 2018?
So this was 2016. Yeah, 2016.
Did you know about the advantages of tissue culture for cannabis at that time when you started? You know, because today, because of these viruses and pathogens and endophytes and all this, we know why it's good for, and preserving genetics, of course. In a small facility, you can preserve like a lot. So, did you know that at that time when you started it?
Yeah, I did not. You know, one of the realities is an academic mind and an industrial mind might be different, even though they both at the fundamental stage are trying to solve problems. The problems that they might be trying to solve are different. In academia, tissue culture is simply a means to an end. You almost exclusively use it to propagate organs or cells, like mammalian tissue culture, bacterial tissue culture, fungal tissue culture. Mostly is done post genetic engineering, because you have a cell that is very easy to engineer. You can't necessarily do genetic engineering on even two cells, because if you only successfully do one, you have a chimeric organism. 50% is genetically modified and 50% is wild type. So you have to go down to one cell, the base quanta of basically living biology, and then you need to remunerate your organism. So you do tissue culture to get that one cell to many cells, to an organ, to a complex tissue, into an organism, right? That's always how I had been taught about it, had seen it utilized in research laboratories, in academia, and then in industry, it was radically different. Now we're talking about growing high densities of monocultures in enclosed environments. So the risk of communicable diseases getting in there and spreading is much, much higher. And now using tissue culture as a means of putting this potentially sick plant into a pocket of sterility, cleaning it in that pocket of sterility, and then regenerating a clean plant is a unique to industry pursuit. It's not necessarily one that you see commonly, and personally not one that I had ever seen done in academia. So it was new. It was a new way to think about it. Fortunately, the technique and the steps are the same, whether you're reconstituting a cell or a meristem or nodes or whatever. The timeline might be just a little bit more extended.
Is the reason why it wasn't as used with other plants because more of them are seed grown?
No, so I don't know if I would say less used. I think the thing is,
I meant as far as like getting rid of virus and disease. Or maybe I misunderstood you.
yeah, I think the thing is most other plants have alternative strategies that are allowed in their industries or accepted in their industries. I mean, one of the things is like the easiest way to get a PM resistant plant is to find a single protein that PM uses to latch onto the plant and either change its conformation or remove it entirely. And now you don't have any more PM, and now you can just spray PM on your plants and they'll never get on. You don't have to spray any pesticides, anything like that, right? But I think the cannabis community in general has certain negative attitudes towards GM crops, and that's totally within their respective to hold. So within within being, again, an industry lab that has to be aware of people's attitudes and pressures, You want to do what is most acceptable in the industry. So tissue culture has been forced that way. I think if you look at how to remediate like Hopsa and viroid, which is not necessarily spread to a lot of other plants, but there are other viroids, much to your point, that have spread to other plants. They are usually remediated in other ways. And maybe sometimes they do use stuff like meristem and thermal combinatorial methods, but oftentimes they'll use something different that we're just not using in the cannabis industry.
Like a chemical application?
Yeah. Or like as mentioned, like genetic engineering crops are very common. So in America, you're not allowed to buy directly genetically engineered crops. That's slowly changing under the last two administrations. But like genetically engineered byproducts are very allowed in food. So go pick up a Snickers bar. It'll say right on there, made with genetically bioengineered or whatever terminology they're trying to use. That's more accepted in their pursuits. I think cannabis has a very holistic attitude, and we should be really respectful of that. And so part of that leads to meristem is the industry accepted way of remediating something like Hopsa and viroid, and it works. So
before we get too much into that, I wanted to ask you about, you mentioned ethics, and I'm wondering when you went to the university, if this was a question of them, like, was there any, you said Professor Berkowitz, immediately was hip to the idea of doing this study, but was there any pushback from any of the other members of the university?
So I'm sure if I really thought about it, potentially, right? Because similarly, the university is not a monolith. Many of the professors come from different countries that have varying different attitudes, some even more positive and some potentially more negative towards cannabis, drug research in general. So it'd probably be naive to say there wasn't any pushback. To say that I experienced any of it I can certainly say no. Like, I was in the plant science department at this point. Pretty much everyone I knew in the molecular biology department smoked. A lot of the professors smoked. It was not certainly talked about very readily, but there was a moment where one of my best friends, who was doing his graduate program at the same time as I, his professor was about to publish, and his protocol for publishing was to basically go home, lock himself away for 2 weeks to write the whole manuscript, and just smoke like a pound of weed during that time. So he asked me to help him procure the pound of weed for the professor to go, because I was in the program, and I'm like, as many times I have helped people, this is hemp. It's not obviously what the program is for. The professor got what he needed. So my experience with it was almost like everyone was fascinated. Like, everyone is doing fascinating research on this illness, that plant, this organism, whatever it was. But we were kind of doing the cool thing, the taboo thing with cannabis. So a lot of people were like, what are you guys going on? What kind of how are you guys going to tackle this approach? Let me give you my 2 cents. It was very, very enthusiastic.
That's cool.
Yeah.
And so from there, when you started to have success, you made your own company, Kinomic?
Yeah. So when I left UConn, I had already had some very positive industry connections that I had talked to just from being with UConn. A lot of people in the industry, for example, like we talked about funding, because at the time, and I mean still technically is, cannabis is Schedule 1, a federally illegal substance, and UConn is a federally funded school. If we were to use federal funds on cannabis, there was a risk or a concern that all federal funding for the entire school, every unrelated department, from psychology to puppetry to everything that got any federal funding, would get their federal funding pulled, obviously in addition to us, the Plant Science Department.
That's why I asked the question about ethics and pushback.
Well, so that's fair. From a funding perspective, we couldn't write a traditional grant and go through that type of process, which is what every other laboratory in that department was basically doing. we had to go and find industry partners. Connecticut at the time had Curaleaf, which is a major MSO. I think that was pretty much the only MSO at that time. Since then, a few others have moved in, but they were essentially like angel investing and funding this. And that was like the only way we could get it to go forward. We were very fortunate. They literally never asked anything. One of the nice things about this is it's a pretty niche, nuanced science. So it's hard to make demands like you might expect an investor to do if they don't necessarily understand what's going on. They're more just like excited that the research is happening, and probably realistically, it's a mechanism for them to say like we are supporting research and stuff like that. They never asked us what our progress was. They never asked us what we were doing. They were just very kind.
So by angel investing, that means that they weren't trying to make a profit. That they were doing it because they believed that it was a good thing to do.
Yeah.
And,
I can promise you they made no profit. They exclusively hemorrhaged money into that lab. And then again, because public university, everything, even though it was still privately funded, everything that was discovered and published is published in public, journals, not given to them, exclusively, stuff like that. So yeah, they exclusively lost money. I think this was just a check that they were comfortable to write and then forget about.
Must be nice to have that type of money. Hopefully someday I will, and I'll be able to be an angel investor for good causes like that as well. But I mean, it's an incredible thing to think about.
Yeah, we were incredibly grateful.
And so I wanted to just take a step back and say, or ask, the company that you used to work for in cannabis, they told you, or they asked you if you would look into tissue culture, and you went and did that, but you never returned back to that company?
Yeah, so when I was there, I was incredibly grateful for the opportunity. They initially brought me in to help in the lab, in the extraction laboratory, but had asked me to, one day do tissue culture. And I was like, great. I know how to do that with bacteria, fungi, all the stuff that I've done in academia, but not plants. And that was what led me, as I mentioned, to asking Dr. Berkowitz about his lab and stuff like that, and then him asking me to work there. When he offered that opportunity, it was incredibly hard to think about returning back to this facility and knowing that, like, while my opportunity to grow in that facility and in that company was high, and I believe that they have a really good corporate structure and they promote from within, so it was really tantalizing. My capacity to learn would probably have never been as high as if I had stayed in school and just committed a few more years. And ultimately, that's just what I decided to do. I think for a very short period of time, I would help out at the lab and then do schoolwork, but ultimately stuck with school, and I'm very thankful for making that decision back then.
So that was another question I was going to ask, is you were still considered a student. You weren't like working for the school. You were a student, and you were...
Yeah, he was a graduate.
Right. Okay. Nice. And so, at what point did you stop this endeavor at the school and take it to the next step?
So in 2018, basically, finished up at UConn, and then was very poorly planned. I mean, I knew I was leaving. Everything was set in stone. I had met with all my committees and stuff like that. But planning, job hunting, all that type of stuff, I was so focused on finishing all my research and stuff like that to make sure that I could just clean slate leave. It was a little short-sighted. So I got fortunate to help someone with their grow operation, and that provided the nest egg to essentially start Kinomic. And as soon as I could, we started it. So within 6 months, we had some operating agreement, we had a lease, and a dream. We were off to the races.
And when you started Cannomic, Was the plan to do tissue culture for others, or to be like a genetic repository, like
Sure.
you guys are doing with the Grateful Head, or?
Sure. So initially, it was actually just to do classes, and that's basically what we did for the first 2.5 years. We were just offering tissue culture classes. Come in, we'll show you how to do micropropagation, we'll show you how to do meristem, make media, whatever it is, right? Synthetic seeds for a while, and then at one point, the classes were a little bit of a premium because self-funded self-funded lab. So the people that were coming into the classes were people like Skunk VA and
I was going to mention him.
so incredible individuals in the industry. And essentially, at one point, I was looking at the people that I knew, my Rolodex, for lack of a better term, and was realizing that it was an incredible list of individuals, like an incredible culmination. Um, so once that realization happened, the opportunity to expand beyond classes started to conceptualize, forming a network of people that really understood the plant, really understood the legacy of the plant, especially within the community, and the genetics of the plant itself was just, again, yet another opportunity that I could not afford to let slip by. So, for a very long time, we had genetics that were just our own. We never really worried about it. We would usually just tell people to go to other fantastic companies within the industry if they were looking for stuff like that, and we were just focused on education.
So you weren't even doing lab work for other people, like multiplying their plants or cleaning
No.
their plants in tissue culture. You were just doing the classes.
Yeah, we did consults. We helped people establish their own labs. Um, but yeah, like we were doing a lot of tissue culture work for no one but ourselves, essentially. And essentially, like, again, like in academia, to prove something, you really just need 3 successful plants to show that it can be done, that you can replicate it. That's what the second plant, and that you can have an average. That's what the third plant does, right? In industry, you need to be like 95% efficient. So getting that level of efficiency, and again, really just a mind shift. There was a lag on me personally. So eventually, industry mind kicked in, and the opportunity that we were sitting on finally clicked, leading to where we are now.
Yeah, the Grateful Head. Yeah, when I spoke to Jason VA Skunk about you being on the show, he was quite excited, and he's like, "That's my teacher," and
Jason's a great guy. I wasn't
Yeah.
going to name drop him. I never know how he likes to roll, but
I think he's out in the public now. People know that he goes by Jason, and frankly, I think I've outed him already a few times. So he's
well, I'm
out
happy
there.
to ride on your coattails and say that Jason is a fantastic
But
human.
nobody knows him as Jason, so even when I say Jason, I always have to say Jason VA Skunk. That way, everyone really knows who we're talking about. But yeah, he's been telling me about his tissue culture journey now for a few years. When did he study with you? Do you remember?
This is going to be a bad clip, but I believe he took it in 2022. Actually, I think I know exactly when he took it. I think he took it in June or, oh man, going backwards in a month. May is before June. I think he took it one of those 2 months, because after that, I took a extended holiday, and when I came back, our first month in September, our neighbors, their facility caught on fire, and they flooded our facility because our sprinkler system went off with the smoke, which prompted us leaving Western Massachusetts and moving to Maine. So I believe he was in the last successful class, because quite literally that fire happened during a class. So we had to do Meristem Day at my house, which was exciting. But he took that, I believe, and if not, Jason, I'm sorry, correct the record.
I don't think it matters that much. like I said, I had talked to him like after him doing your class, and he's setting up his lab, and he's so excited about it. And I'll take a minute to tell a little bit about my tissue culture journey. And we spoke about it a little bit before we recorded. And we mentioned Bill Graham. He made the home tissue culture kits.
Microclone.
And what'd you say?
The microclone kit.
Yeah, the microclone kit. And so my funny story about the microclone kit is, first of all, I knew Bill Graham from before he ever was doing, the microclone kit. He owned a hydroponic shop in San Jose. I think it was called Pure Foods. And, he also did some wholesale distribution. So me being in the grow shop business, I knew him for a while. And I remember also we went to Epcot Center, and at Epcot Center they have this place called The Land, and in The Land you could buy tissue culture plants. It's actually the only place that I've ever seen that you can buy plants like in vitro, in the gel. They'll sell them to you there, and we bought some redwood trees, and I don't remember what else they sold. The redwood trees was the one that I really remember. And after that, Bill had his microclone kit, and I was super excited about it, right? So, I bought the kit, and I think that this is very similar to what a lot of my clients did. Was I bought the kit, and I did nothing with it for years. And the reason why I didn't do it was because I felt like I needed to be ready. I needed to make the space. And I really had to just get my head around the idea of doing it. And so literally I kept the kit for over a year. The ingredients might have already been bad in it. And, one day Bill came to my store and he said, "We're just going to do this right now." And I'm like, "What?" And, I had a retail store. And we did it right in the middle of our retail store with the front door opening, with a Rubbermaid Tough Box turned on its side. And we divided it up, some redwood trees. The other thing that we divided, I don't know if he bought that at Epcot or if it was something he had, was scented geranium. We divided some of those. And unbelievably, it all worked. And there was no contamination, like zero. I don't know if he puts like preservatives into his system or want to make it so that there's no contamination, but for someone that had no experience using scalpels and forceps and the front door opening and just working in this Rubbermaid Tough Box, to see that actually worked was really inspiring for me. And so at that point, I started doing tissue culture right there in the middle of my store every day. And I had 2x4 grow tent that had 4 shelves on it. And that thing was just packed with jars of plants that were growing. And I even shared a picture with you, and I'll put it up in the video of and this is one of my shockingly proudest plant moments, scented begonia was blooming in the jar. And I just think it was so incredible that happened. The redwood trees were super easy to grow. They'd grow, we'd cut it up into a million pieces, put it in there, and they would just grow. And so before you knew it, I had this tent filled with tissue culture plants. At that point, I still hadn't ever done cannabis. And I was able to inspire people to purchase the kit. Okay, and we got, I don't know, maybe a couple of dozen people that purchased the kit, and no one used it. For the very same reason that when I started my tissue culture journey, well, there was actually 2 reasons. Number 1 was the intimidation factor, right? The other thing that stopped people from doing it was they didn't get it wasn't helping them now. I told people, hey, look, if you do this tissue culture, you'll be lucky if you have plants that you could take out of tissue culture and actually use in your garden in 6 to 9 months. Because the sterilization process is super stressful, then they got to grow, and then you got to divide them before you do it. And so because they weren't getting, I don't want to say immediate satisfaction, but immediate benefit, they never did it. And like literally, I can say with confidence, 0% of the people that I sold that kit ever used it.
Sure.
And so one day I went over to my buddy's house, just like Bill Graham, and said, that's it. We're going to do it. You know, because I wasn't growing cannabis at the time when I had my retail shop. And so I said, you know, we're going to do this. And we did nodal tissue culture. Of course, wasn't doing meristem. And again, it worked. And the plants kind of looked a little bit weird, which is something that I wanted to talk to you about. They were like a bit glassy. The leaves were like, you know, like shiny and very transparent, and even looking a little bit fragile.
Sure.
But they grew.
Yeah.
And, you know, the mistake that I made, and I've actually made this mistake multiple times, was I didn't take it to the next step. The plants would grow into the top of the jar, and eventually they'd start to get damaged because they needed to be, you know, moved. And yeah, that's pretty much where my tissue culture journey ended. I never took those plants, divided them, although I know that I had already accomplished the hardest part, was sanitizing the plant, because, you know, I rarely got contamination, but it was common that the plants would get damaged in the sanitization process, and they would just never survive, you know, putting them into the gel. But I never, like I said, I never divided them again, and at that point, I wouldn't have needed to sanitize them again, because they were clean, they were living in the jar for some time, nothing is growing in the agar, so I don't, you know, so it was just, again, yeah, at this point, I wish that I would have taken that next step. And what I wanted to say is, in talking with Jason and hearing about the Grateful Head, and also we have another host on our show. He goes by Cosmic, and unfortunately, he doesn't make his living in cannabis. You know, it's just his passion. He has a home tissue culture set up, but he does meristem. And, you know, he learned how to do this on his own by, like, reading books and YouTube videos and stuff like that. But he never used hormones. He just puts it, I believe, in the agar, and I forget what the stuff is called, muskage or something like that.
Murshy and scooge. Yeah, MSG
Right.
is what most people call it.
Yeah, but yeah, he doesn't use the hormones, and the plants grow from a meristem sample into plants. So I guess now that I told a little bit of my story in tissue culture, well, before I do, as I want to say, is I'm re-inspired to learn more about tissue culture because of Cosmic and you and Jason, VA Skunk, and what the Grateful Head are doing. Now I want to continue this journey, and I have a little spot at a friend's house that he's built a tissue culture lab. I'm going to share some pictures with you and with everybody. So I want to learn about it, not because I want to make it into a business. I mean, maybe that'll happen someday, but more because I just want to learn about the process, and I want to be able to preserve my genetics. And
all right, so still gotta like just wipe the floor up a little bit, but this is our little like kind of scuttle room to change in, and right here we'll have we got a lab fridge set up. There's gonna be a rack for tissue cultures with a bunch of intelligence to call me if there's problems. Laminar hood, got the mini extra laminar hood for doing the meristem stuff. yeah, all the tissue culture vessels and shit, and I'll be able to fill this all up with like 200 ball jars. So that'll be our starting next week, I hope.
You know, while there are other people doing it, you know, there's places that will bank your genetics. It kind of becomes expensive after a while, you know, and so for what it costs to bank some genetics, you can, you know, learn how to do it maybe. And,
Sure.
you know, and I also like the idea of being in charge of my own destiny, you know, so I want to learn about tissue culture. And I noticed, and, you know, we'll tell everyone that Philip is still doing the classes. And if you go look at his, I'll include the link on the description, but on the Cannomic Instagram, he has a link tree that takes you to his website where you can see the services that he provides. So,
Sure. More than happy to, except I'm going to do something totally different. I'm going to address a bunch of the stuff that you said.
Okay.
So the glassy leaves, that's a classic condition called hyperhidrosity. It's quite literally, these plants are in an environment that often when you put them in there, the agar's been poured, it's cooled and dried, and the environment is dry, right? So you put a plant in there, the agar is mostly water. So in this open wound, the plant is going to start pulling water from the agar, and any nutrients, hormones, whatever else is in that media mix will come into the plant too. And then it will start transpiring the water out of the leaves as it produces new leaves and stuff like that, right? And you'll eventually see condensation form in your container because it's essentially a humidity dome. But humidity domes often allow the humidity to vent. In a tissue culture, because the container is often fully sealed, maybe a small vent at the top, you'll reach an equilibrium where there will be saturation of water in the air, leading to condensation, just like it does when you take a hot shower on your window, on your mirror. The only reason that's happening is because the air has saturation, so it's looking for somewhere else to stick, and the walls of the container aren't easy to do that. When you use, especially some hormones like cytokinins, and especially TDZ, which I know is very commonly used in cannabis tissue culture, it will promote hyperhidrosity. It essentially forces the plant to keep sucking.
Did you say TDZ or TDT?
TDZ.
Yeah.
Thidiasterone.
Okay. Right. That was one of the
I'm very
one of the
familiar
kits
with
that I got.
Yeah, I'm very familiar. And so what it'll do is, like normally under regular biological regulation, once you reach saturation, plants will stop pulling in water and stop transpiring water, which will slow down their growth because all of the nutrients and all the hormones are in the water in the agar. It's a hydroponic system. So the more water they pull through and push out of their leaves, the more nutrients, the more hormones are getting through them. Some hormones can force them to do it beyond the regular regulation. This is basically what hormones are designed to do, are to override regulatory. Just like adrenaline overrides your regular normal state of being, but then very quickly has to go away because you can't live your life on adrenaline. We'd all die of cardiac arrest in our 30s. Something like cytokinin, especially TDC, will continue to force the plant to pull in water and push it out, which leads to hyperhidrosity. It's beyond a saturated level of water, and the plant morphology especially looks incredibly glassy, not very good. Venting helps that. Lowering your plant density helps that. Just as two very easy ways.
Yeah. I wasn't venting when I first started. That, you know, I didn't even think that was an option.
Sure.
And the last time I did tissue culture, which was a year ago, I did buy some little stickers that you could drill a hole in the top of your container, and then you put that sticker on. And I mean, I'm assuming that it was a combination of both things. That first is that I was venting them, but also at that point, Bill Graham had like multiple different kits. Some that had TDZ, and then some that had other hormones in there. And you know, this last time that I did it, I didn't have that problem of the glassy leaves.
Makes total sense. And then another one
Sorry to
that
distract you.
no. You're totally good. My ADHD rattled mind is never going to be so good at one topic. But I remember you also mentioned when the plants would hit the upper threshold of the containers, right? So plants have no natural defense or strategy for an upper limit. Like that they're the tallest growing thing in every environment, except for shade crop. And shade crop grows radically different than a tall growing plant like cannabis, right? So tall growing plants like cannabis, they have one plan, right? They hit something, they try to bend and grow around it. It's basically a gibberella ends up work. And what you probably saw is they'll hit it and then they'll start growing like this. They're trying to go around, they're trying to sense it. Eventually, when it realizes it has nowhere to go, it will self-top. It will literally necrotize its own apical node and let the lateral nodes start shooting off because maybe they'll have a chance to bypass this upper threshold that is preventing it from growing, right? When those lateral nodes hit the top and they also die, usually the whole plant just has no backup game plan. And that's probably what you saw. You probably saw it hit the top, this top part just kind of die and look sad, and the side growth, just like a plastic top would go. But then eventually the plant doesn't know what to do anymore. And then finally, what I'll say is the hardest part of tissue culture is always going to be organogenesis, growing a new organ out of your plant. So for nodal micropropagation, that means rooting. Your plant, I mean, think about traditional propagation.
So not growing without roots?
No, that's super easy. The easiest thing in the world, right? So think about like a classic plant that you have right now, a veggie plant, right? You cut it, you dip it in Clonex. 10 days, 14 days from now, there's going to be roots at the base of the plant, right? Where did that primordial, original root cell come from? Where did it come from? Where did that initial root cell that made the second root cell, that made a clump of roots, that little white nodule that eventually made the full-grown root, right? How did that happen? I mean, that happens every time. I've never seen your gardens, and I can guess that's what happens because it's not coincidental. It's mechanistic. What's the underlying mechanism, right? The underlying mechanism is that you are changing the cellular identity of those branch cells at near the injury site. And you're telling them to stop being branch cells, become stem cells, quite literally callus. And out of that callus, because callus naturally wants to bias towards roots, they root. All under the direction of IVA, right? The active ingredient Clonex, NAA, IAA, whatever auxin you're using. So in tissue culture, if you don't close the wound, you can just keep it going indefinitely. Because you're not growing new organs, you're just letting the organs you have keep producing new ones. You have already a node, so the node will grow into a branch, which has many nodes, which can grow into many branches, which will all have many nodes. Growing the roots out of it is the hardest part. It's why it's the only part of our entire growth cycle where the plant doesn't grow at all for 2 weeks, just freaking chills while it commits all of its metabolic energy to generating a new organ. And then once it has that organ, it starts growing, and then your clone grows, and then your veg grows, and your flower explodes in growth, right? But your clone, before it roots, it's not growing at all.
And what you said, Phil, was really interesting, that growing the roots is the hardest part. Mm-hmm. And I agree that, when we're not thinking about tissue culture, one of the most magical things that we do with these plants is take a cutting and get them to root. It's a pretty incredible thing to do and make happen and multiply plants. But for someone and for people that have been cloning cannabis for many years, mm-hmm. maybe it's not the most difficult thing, but the most amazing thing for us to see is a plant growing without roots.
So what I would say classically is like tissue culture is one of these relatively new techniques, right? Once, like you're saying, anything that isn't normalized is magical. And once it becomes normalized, it's just science because it's understood, right? So like if you were at a party in the year 2000 and you had an aerocloner and you were telling people you cloned in the air, they'd look at you like you were lying to them or a warlock, right? One of those two. But now if you tell people you clone in the air, they'd be like, yeah, I totally understand because I've seen a lot of air cloners. It's no longer magical. It's just science. Tissue culture is the same. Like thinking a plant can't survive 20 minutes in bleach and a little bit of soap and then being put into pressure cooked media is fantastical because it's foreign. But when you do it, much like cloning, it just becomes a job, right? And then, like to many people who pursue gardening as a hobby, they have a radically different relationship than people who pursue agriculture as a profession, and they're growing thousands of plants rather than a couple of selectable, right? So, I think every part of tissue culture is fascinating when you do it initially. Similarly, seeing the roots in a clear media that you can just flip over and your plant doesn't fall down and you can see them growing out of the spiral. You can actually see the initial injury still and see it growing because it's not obstructed by a rockwool cube or some kind of plug. I think that is in itself quite fascinating. And then I think at some point people are just like, okay, on to the next one.
Right, of course. After you've seen it for a while.
Yeah.
For me, still seeing the plants multiply itself with no roots. Over the course. I mean, just seeing a plant in a jar is incredible.
For sure.
It's like you have that like the movie The Biosphere or something where people were living in this jar and like, particularly before I put the vent on the container, it's like nothing goes in, nothing goes out. It's just an incredible thing to experience, and as you see it much more,
So when we,
I just thought it was interesting that you said that the rooting is the hardest part.
Only from a purely biological standpoint, growing what you have already is pretty easy. You just got to keep on eating. Growing something new is incredibly hard. So, and then I like to relay it because we've all cloned. So we've seen our plant just sit still for 10, 11 days until it roots. And then once it roots, it does what the plant did before I cut it, and after it roots, it just grows, right? So I only want to relay that. I think you're totally right. When we go to conventions, when we go to shows, we bring very pretty rooted plants in the containers because when we move them out of the container, everybody knows what a plant in veg looks like. They have them. But in the container, if we're going to a show or let's say a convention and someone else is selling genetics over there, it's very easy to be like, well, if let's say their genetics are clean, but someone else walked in with potentially russet mites on their boot or whatever, they can have access to those plants. At least our plants are in completely enclosed vessels, so that provides a level of peace of mind that is hard to find elsewhere. And people are incredibly fascinated by it, way more than the science. They just like the plant in the container, and that's totally okay too. Not everyone has to know how something works to appreciate it.
Well, I mean, that's what you said, is people don't know how it works. But like when you're in school, you learn that the plants uptake nutrient from the roots, and they bring that up, and then the plant photosynthesizes and creates more foliage and shoots and maybe fruit. So it's just taking that step out of the equation that leaves people going, hey, I learned that this is the way plants grow, that they need the roots.
Yeah. I think what happens is, this is very common. I remember like in an early class, we often hear stuff and we fill in the gaps, right? Like, I remember being in an early physics class, a little bit off the beaten path, and there was like, okay, two cars hit into each other. What do you expect to happen? It's like, what you expect to happen is they both bounce back, right? It's like, that's exactly what happens. Now a heavier car, and they both bounce into each other. The heavier car, you expect to not go flying backwards, and the lighter car to go flying backwards, right? But explain why. Explain why, and it's incredibly hard if you can't explain the concept of inertia. But you fill in the gap, right? You fill in the gap. And then it's like, well, what happens when I run into a wall really, really fast? I bounce backwards because I have less inertia than the wall. But people don't think about it this way. They fill in the gaps. I think the same happens with like tissue culture or anything else. This is why I often try to, in my classes, relate it to conventional or traditional propagation, because it's something that we're much more familiar with. And fascination is good, but it's a science. We should be demystifying it and helping people understand it So that way, like you, if you want to pursue it, the more demystified it is, the easier it'll be. Like, imagine how hard it would have been to even do it on your own if Bill had never come by and offered that initial assistance, right?
It never would have happened.
And it's probably maybe because it seemed fantastical and very mystical.
It was intimidating.
Exactly. So I very much relate. Or am empathetic to people like that. That's why we thought the classes would be as popular as they ended up being, because people do really want to know. And even if they don't do it, they at least want to understand what other people are doing to make the most deliberate decision for themselves.
One of the main reasons why I wanted to do it back in the day and still want to continue my tissue culture journey is it's very hard to keep lots of genetics.
Mm-hmm.
You know, I mean, honestly, if you just have one, it can be hard to keep that as well. We've seen many people, you know, including me, lose valuable genetics. And so when Bill showed me, and I just love the man. You know, whether his kid is perfect or not, I just love the man. So, because he inspired me in a lot of ways, not just that. But what he showed me was, hey, look, in one flat, you can have 30 jars. And even if you have redundancy, let's say you have 5 jars of, you know, one genetic, okay? You can have 6 genetics in a flat, okay? And then you look at how many flats can you have in, you know, this 2x4 tent that I had with 4 rows on it. It's like, this is a way for me to preserve so many different genetics.
Yeah.
You know, where like, I think what the Grateful Head is doing is really great as far as they're holding these genetics and providing it to people. But, and this is nothing negative to say about the Grateful Head, but like, there have been other companies that, you know, people have paid to store their genetics and they went out of business.
Mm-hmm.
You know, and so like being able to do this stuff yourself to me is attractive because, again, you're in charge of your own destiny. You know, something that we talked about, you know, on many other shows is like sharing genetics, and, you know, part of the Chem Dog story is, you know, how it was shared, and if it wasn't shared, it wouldn't be here.
Mm-hmm.
So, I mean, using another place to, you know, do tissue culture as well, and giving cuttings to your friends and people that you know, I think has value. But, you know, one thing I want people to leave, or I'd like people to leave this show with, is an inspiration to, you know, learn about tissue culture and see, you know, because even though I didn't follow through, you know, for one reason or another, you know, things happened and, you know, I wasn't able to complete my tissue culture journey. I'm going to continue it forward. The benefits of growers using this technology, even if it's just nodal, you know, for instance, if you don't have viruses, if you know this, right?
Sure.
Let's say you purchased your clones from Grateful Head and you know that they don't have the virus. Then just keeping them and dividing them using nodal tissue culture is a great way to go, you know, as far as like the amount of space that it's going to take. So I want to encourage people to, you know, take a look at, you know, not only Bill's class, you know, kits, but Phil has these classes. Now, do you sell kits as well, or do you just do classes?
We just do classes.
Because the ingredients are available.
Yeah. I mean,
right?
Yes. I think, like, basically what Bill taught you more than tissue culture is that it's technique over everything, right? You didn't need a huge setup. And our first lab when we started was in a basement of a New England mill, probably one of the dirtiest places in this world. And, like, it didn't matter because we didn't have to be sterile everywhere. We had to be sterile in specific moments, right. in out of the autoclave, in the laminar flow lift. Everywhere else, it's all capped, right? So the same reason I can bring it to a show is why I can do it here or there. I want to, like, I want to just, like, adjust it in a pretty simple way. The more self-sufficient you are across the board, it's not a tissue culture specific thing. The more self-sufficient you are across the board, the better. if you have land, it's good to learn how to grow agricultural crops, food crops, even more than necessarily something like cannabis. Something like, you should learn how to grow food. You should learn how to do basic maintenance on your car, basic maintenance around the house. Like, all of this is better than calling in a handyman. And it also makes it more applicable that you'll know when it is appropriate to call in a handyman versus like switching out a fuse yourself versus necessarily needing to rewire something because it's a deeper issue than that, right? So knowledge is never the wrong word. It's always good to be as self-sufficient as you can be. I think it's ultimately comes down to convenience and price, right? Like, there's a reason why an oil change is one of the easiest things you can do on your car, and it's one of the least common performed tasks that people do because it costs $30, $40 for someone to do it, and many people would rather the peace of mind of having a professional do the oil change, and if they mess up, whatever issue, whatever liability that comes is on their hands, rather than if I mess up an oil change and now my driveway has a beautiful rainbow sheen for the next 6 months as the oil just kind of stays there, I have to deal with it, right? This is always like, if the company goes out of business and they lose the strain, there honestly might be like pathways that people can take to at least get some level of I'm losing a word, but like to get some value back, right? There may be like legal pathways and stuff like that. If you lose your strain in your tissue culture lab, then it's like, that's it, it's gone. So the best case is like to do it in tandem, ideally, right? I mean, it doesn't have to be some paid facility. If you and your friend both learn how to do tissue culture, backing up each other's plants off-site of each other is always going to be the safest way to ensure it. It's like you're saying, it's the reason ChemD survived was because everyone had the ability to keep it going, and its provenance was never questioned. So we all know what it is, and everyone wants it. It's great, right? I highly recommend. Like, I wouldn't be able to do tissue culture if I didn't learn how to do tissue culture. None of us would be able to grow cannabis if we didn't learn how to grow cannabis. Even if our parents were the greatest cannabis growers ever, we would still have to learn for us to be able to do it. Tissue culture, and much to maybe your lived experience, it's not rocket surgery. It's not the hardest thing in the world. Meristem can be very tough. You need a little bit of a microscope, and working under magnification can be tough because your own movements are magnified. So your movement to the left is 30 times more to the left now under a microscope. But otherwise, it's just muscle memory. That's what growing is. That's what we all have. So I highly encourage anyone who has any interest to learn it. Nodal's very beginner friendly, especially if you have any experience with cannabis cultivation. It's very similar to cloning, just on a much smaller scale.
And something else that always concerned me about tissue culture, that I think was part of the reason why I never made the full-on jump to it, was I was scared that, okay, I can have these genetics in tissue culture, but because it was still foreign to me, I felt like I still had to keep the moms. Like I was scared, like, man, like these things, they could get contaminated or they could die, and then I lose the genetics. But I know that if I keep these mother plants, I know how to do it.
Yeah. The golden rule for data backup is 3-2-1. 3 copies, 2 different medias, 1 off-site, right?
Right.
So 3 copies can be you, your buddy. One of yours is in tissue culture, one of them is a mom, right? It's an analog backup and a digital backup, for lack of a better word. And then your buddy is the 3rd copy, and he's off-site. So if anything happens in your facility, you can always get the genetics back. I mean, you know how to do this.
It's hard to do that, particularly when you get more and more genetics.
Yeah, I mean, space is ultimately the name of the game, right? I mean, I think that's where, for many cultivators, you clean up your plants only once. If your SOPs are good, you don't necessarily need to be doing meristem cycle after cycle. You should just have a clean mom, and then now you have a clean mom. Stop getting it dirty. So for many cultivators, it's exactly what you're describing. It is a space thing. They have sentimental or non-commercially viable but still valuable plants, and they need some way of storing it I mean, I still recommend keeping a mom around, universally. Like, you.
But that takes away one of the big advantages of tissue, like that I mentioned, of tissue culture, is that you're able to, keep so many genetics in a small space.
Sure.
Well, if I have to do that,
Yeah.
and then still have another space to keep the moms, then that whole advantage
Well,
was
I would
ruined.
change it, right? Because right now, let's say you've got 3 strains, right? So you've got multiple moms per strain, usually at least one's maybe a little bit smaller, keeping the lineage going, and one is being cannibalized for the round of cuttings or propagates that are being made, right? In tissue culture scenario, you would keep a single mom alive, literally just to be the backup nodal donor if it comes down. And ideally, that mom came from a meristem plant. So you know it's clean and you never cut it. So it's never going to be introduced to anything. Nothing's ever getting in there. It's just in a corner somewhere growing, never needed. And maybe once every other month it gets cloned down or a fresh one out of TC comes out to replace it, right? But in tissue culture, you are multiplying. So you are making more copies. That's your cloning step. And you're rooting. So you're producing clones. Like, you are basically replacing the entire mom stage. In tissue culture, you would just keep a single mom as an analog backup. So yeah, it's a little bit it's a little bit of a larger footprint per strain. I think it's still considerably smaller than what many facilities are running now. Like 5 moms per strain, so that they can meet their threshold run.
How many clones they need.
Yeah, exactly, right.
Right.
So throughput in tissue culture is very, very easy. It's the main thing you see it being used for in industry outside of cannabis. Like, commercial, agricultural tissue culture is purely for production. So like seedless watermelon, they're great. Love them, right?
Okay.
Seedless grapes, whatever. They're seedless. You don't get a second generation from them. They're not going to give you any seeds, right? So the easiest way to propagate them in the high density that's needed to reseed all the fields for the next generation is through tissue culture. And seedless watermelon, all the plants that you can buy at your local hardware store, if they sell them like mums or stuff like that, at Home Depot or whatever, all of those start from tissue culture and then eventually make their way out, right? So for propagation, cultivation scale, it's an immaculate tool for like this large scale cultivation.
Yeah, for sure. Sasha, I know I interrupted you before when you were asking a question, so go ahead. We both have so many questions for Phil.
Yeah.
Hopefully we can...
Actually, I have to tell you, I have a facility of mother plants also here in Austria. It's next to Slovenia where I live, but it's the same thing for me. Like, I and Gaby, when we met, we started to talk, and both of us have a lot of similarities, and one of those is definitely the one that we are both dealing with the thoughts about starting a tissue culture technique, because we are getting older, Mm-hmm. All of us in this industry, we have to keep up the genetics for years and years, and you are sometimes the only one that has a particular cut. You're actually begging other people to keep it also, but it's hard, it's not, so something that is maybe only valuable to me right now maybe will be valuable in 30 years to many more, like Gaby mentions the chem, that's exactly this kind of story. And the thing is that you realize when you work with plants a long time that there's a particular cut that you work with every time. So if you keep it like really in the perfect shape, and the traditional cloning works, right? But the problem gets when you start to get this kind of, when the cut's getting like 30 years old, mm-hmm. And then you can keep it up just for a certain amount of time. And then, because I was listening to Skunk VA's podcast last time, he was on a podcast, he told about these endophytes, right? Maybe it's not the virus, the Hope latent virus, but there is this so much other stuff, right?
Yeah.
And I wanted to ask you before this question. If I cut from a healthy mom, let me say healthy, not Hope latent virus, but it has all kind of other stuff, right? Which I don't even know to name. I make a meristem from it, a cut, and I culture it, and I do then these nodal cuts from that, yeah? will it be clean immediately, or is a chance also that the virus is already in the tissue, right? Or
Yeah.
this?
So like the reality is, it's like how much nuance do we want to jump into? Because what does clean really mean, right? Hops latent viroid isn't new. It's just we care about it now because all the other stuff that we already knew about we can handle, right? No one cares about hops latent viroid when you've got russet mites, because russet mites kill your operation right now. Hops latent viroid is a generational, prolonged annoyance, right?
Man,
Powdery mildew.
I have to disagree with you.
Oh, I mean, but the thing is,
The reason why is because like russet mites may be super devastating to your plants right away, but we do have some ideas on how to handle it.
But that's
and
what I'm
that's like what
saying.
the average
No, no,
person
no.
can do it,
All
right?
Yeah, but that's what I'm saying, man. Like, we used to not know what to do with it, right? And so it kills your cycle immediately. Powdery mildew kills your cycle immediately. No one cares about how amazing your veg was when you've got powdery mildew in flower. No one cares about how amazing your grow is when you've got Botrytis fusarium. But all of these, because they're way more pertinent, have solutions. They needed solutions. We needed to figure out how to stop russet mites. We needed to figure out how to stop powdery mildew because there would be no cannabis industry at all. Hops latent viroid is progressive. It's opportunistic. The reason it can thrive now is because we can handle everything else. So now the next kid on the block, like biology, is a game of finding energy resources and dominating them. And plants are an incredible energy resource. Let's say we do meristem, right? It's a classic question, and I know I'm going to get into the weeds into it. Meristems aren't sterile. They are infected by other things. Mitoviruses have been shown to infect cannabis meristems. Have you ever heard of mitoviruses before this, right? No, because who has? Who cares? What does clean mean? Clean means I want my plant to produce, right? Anything that stops my plant from producing means it's not clean. So hops latent viroid is now the newest version of the plant not being clean, even though it's probably been around for thousands of years. It's not named cannabis latent viroid, and it's not called hops active viroid. It's a latent viroid from a different species. To jump species takes a lot of evolutionary time, and to be latent, it takes a lot of, and then to be actually shown, it takes a lot of metabolic energy. Just like for humans showing herpes, it's a latent virus. It hides and only shows up when your immune system is complicated with some other illness, right? And then it goes away. And then, like, hops is exactly the same. It shows up.
It's a fantastic perspective that you have. Thank you for this, because I was thinking the same, and I'm not. I was thinking is I'm the only one that says hop latent is already been there, you know.
Well,
and people look at me and say,
like Gaby,
no, it's not like that, no.
Let me pose it this way: you have a garden that's got hops latent viroid and russet mites, and there's no solution to either. Which one do you care more about? Russet mites. We don't care about it.
It's far more devastating, far quicker, for sure.
And generationally, if you don't get rid of them and you put more plants in the room, they're right there again. So it has the exact same threat of hops, but like in the immediacy. But we can deal with it. We have miticides and sulfur sprays and all of these, from holistic to pharmaceutical grade solutions. And hops does not have that level of solution yet, or sophistication yet. So I mean, literally meristem is surgical isolation. This is like saying, like, you know, we have a blood-brain barrier. Many pathogens cannot cross it. It is the physical barrier to prevent things. Syphilis doesn't care. It gets right into your brain and wreaks havoc. And in fact, because nothing else can infect your brain or struggles, it has free reign. And so the meristem is like, we isolate the meristem. We isolate a lot of stuff. The mitoviruses, nobody cares about, but maybe in the future, when every plant is from meristem, and we've essentially just hyper-proliferated these viruses or viroids, maybe they will cause issues. But right now, much like hops latent viroid, when you have powdery mildew, you consider hops latent viroid to be basically asymptomatic. So I think, like, you know, to go back to Sasha, your initial question, if you go from meristem and you successfully isolate that tissue, you have successfully cleared up the systemic pathogens we currently care about. But biology won't be static. And in 10 years from now, there may be tomato latent viroid or tobacco mosaic viroid, or like literally all of, and the more crops that are grown and the more land mass that is grown with it, this type of cross speciation of pathogens, is more likely.
And a lot of monoculture also in that traditional sense, where there is too much monoculture.
It's
Then
all
it's all monoculture. These things progress in monoculture, right?
Of course. Well, that's what I'm saying with
it's-
tissue culture. Like,
yeah.
this is why people wanted it, because, I mean, you're talking about monoculture, but I understand that, in a lot of European sections, it's mostly either hybridized greenhouse or all outdoor crops, right? In the United States, it's indoor. So imagine indoor monoculture. The minute any pathogen gets in there, it's wildfire, because that pathogen has no natural predators to prevent it. Like, there are no natural predators for russet mites in my house. There are outdoors, but when I grow the plant in my house, the russet mites get in, they are going to run wild. And I mean, same for everything too, right? So tissue culture is invaluable in that sense. And if your pathogens that you're dealing with are non-systemic, nodal is super good. If your pathogens are systemic, meristem is the best we have right now, because it has a meristematic barrier, just like we have a blood-brain barrier, and that will physically prevent certain microbes and pathogens to get in there, from like Fusarium to Hopsite viral. But to say it's clean.
So I guess in the end, it's far better to start with meristem anyway, because you don't know the systemic issues that you have.
100%.
Right?
Every plant we get intake we do through meristem. Why risk it? I mean, you guys probably have a quarantine area, because why risk it already, right?
But even the quarantine area is only going to tell you about stuff that you can see.
That's what I was like. The meristem is the quarantine area on steroids. You're not quarantining the plant. You're quarantining the section of the plant that won't have the microbes, even if it didn't have it, or even if it did have it. And then, because you know that you're quarantining that section, what results from that section is totally kosher, and the resulting plant you can be less nervous of. I mean, you should certainly still test, and testing services are out there to help you. But meristem is the way to start if you run a tissue culture.
But as far as testing, it's kind of like cannabinoid content. We only test for certain things that we know about, right? So we may not be able to test for certain systemic issues that we don't know exist, right?
I mean,
like you're testing for Fusarium and stuff like that, but
what about like Pythium, right? Like, much to your point, the reality is like it's even bleaker.
Oh my God, it's bleaker.
For many things that we can test, I mean, the test is fine. If you've got Fusarium, it's a fungi. It's got DNA. A DNA-based test is very reliable. Anyone who goes onto a reality TV show to find out if they are or aren't a dad doesn't start arguing with a genetics test, because DNA is pretty concrete evidence of an organism, right? It's just like for example, the Hopsite and viroid testing is a lot trickier, but people demand it. So everyone feels like they have to, but it's almost like security at the airport. It's security theater rather than actual security. They're not necessarily...
Probably at the airport it works even better still.
I would argue, like, you could make that argument, that, like, a lot of the testing that's offered is really just to placate the minds of the people who are doing it, rather than to get a genuine test. And then getting a genuine test is much harder said than done too, for something. Again, for Fusarium, for powdery mildew, for russet mites, it's very, very easy to get a reputable test and not worry about it. But some other things that people care about are a little bit harder. And so...
Just to take a quick flashback in time for me, when I used to own the garden spout and people would come in with, some type of plant problem, it was very common that they would say, so common, it would happen multiple times a day, people would say, I think I have tobacco mosaic virus.
Sure.
And we just felt, and the reason why I'm saying this is because I wanted to ask you what you think, but we just felt anytime there was a problem that they didn't know what it was, they were saying it's tobacco mosaic virus. And now I think people are doing the same thing with hop latent.
Well,
if they're not doing the testing, they're like, oh, this plant is, you know.
Gaby, as an amazing judge of character, I think you accidentally stumbling into something closer to a human condition. Like, you are growing a plant. Your plant is growing suboptimally. There are two realities. Either the environment and exterior outside world is messing your plant, or you are messing up your plant, right? And it's much easier for people who have egos and who have like defensive mechanisms to blame anything other than themselves. I just think it's that simple. Like, my plant is messed up. It's never been messed up before. It must be the nutrients that have never been messed up. It must be the soil that's never been messed up. It must be the viroid or whatever. I mean, most illnesses have obvious characteristics, right? Like, there are generalized immune responses in plants, just like we have generalized immune responses with getting a fever, maybe feeling uneasy, going to the bathroom, like that. No matter what gets us sick, those are the first things our body wants us to do. Is to get a fever, feel uneasy, go to the bathroom, right? Plants do the same thing. Whether if they are overwatered, they wilt. If they're underwatered, they wilt. They have generalized responses, and when people see a generalized response, I think they just want to ascribe it to something other than my fault. The reality is probably like it wasn't tobacco mosaic virus. Its infection of cannabis is relatively rare, and if these are like indoor growers going to a hydroponics shop, then they probably are not interacting with tobacco frequently enough to be live tobacco, not like smokable tobacco, but live tobacco that's got the virus. It's not just readily transmitting it, and then depending on what part of the country you're in, it might not even be hospitable to grow tobacco in that part of the country. So there might not be any tobacco for hundreds of miles. From Connecticut, the only tobacco they grow is in greenhouses because they can make the leaves very, very big, perfect for rolling. But they don't grow any smokable tobacco in Connecticut. So if you had TMV here, it would be, quite a story.
So I guess I was, because kind of like hop latent viroid, a lot of people blame their problems on hop latent viroid. Hop latent viroid does exist, right?
Sure.
And I thought tobacco mosaic virus did exist in cannabis, and
Sure.
that's why this had been happening. So just as an education for me in the world, because you just said that it would spread via fresh tobacco, so it can't be spread like if somebody's holding a cigarette. That's not going to spread it.
Yeah. I mean, like,
because I always tell people, you smoke, you gotta clean your hands. You probably should anyway. But for me, it was to confirm, to just potentially make sure that we're not spreading any, virus or disease.
Yeah, I think like there has been a lot of. It was for the public good to tell people about spreading germs and washing your hands, because the public should wash their hands. Like, that's just a good advice to give people, right? But I think there was a lot of a lot of emphasis put about how transmittable certain things are. Like, I remember growing up and watching this video where someone put like blue paint on their hand, and then every doorknob they touch and stuff like that. It's like, yeah, but like, how hospitable to a microbe is a metal doorknob? Like, yeah, they're on there, but are they growing on there? Am I touching that doorknob 2 hours later, and that's now still a living aspergillus? And it might be. And that, that's a very compelling reason to wash our hands. But something like, viruses, they're not, but they can very simply be thought of as like, parasites. So they need other cellular machinery from a donor to work. They can't just they can survive in the environment, but they can't necessarily live in the environment and thrive in the environment for a very long time until an applicable host gets them. Dead tobacco tissue that's been processed and probably been like infused with some glycerin and stuff to make it more smokable and easier to roll. So I would say you probably have a higher risk of like catching. I don't want to go controversial. You probably are very safe. You are very safe. If you have a field next to a tobacco field, and cannabis likes very similar environmental conditions that tobacco likes. So it's not like to say in certain countries where they could grow both, that they're growing both. Yeah, you could definitely see TMV. And it has some very unique variegation, style, morphology. But that's what I'm saying. Like, if you're just like if you're a home grower in the Pacific Northwest, it's probably very rare that you have TMV. If you're a home grower in the great north of Maine, it's probably rare. If you're in Georgia, it's a more compelling argument.
Makes sense.
Yeah.
Something else that I was really interested in, and Sasha, please interrupt me if you have any questions, because I have so many questions I may not be giving you any time. But I saw on your website, and I've heard on, so I'm part of a tissue culture group on Facebook.
Nice.
And it's all like regular plants, not cannabis.
Mm-hmm.
And I'm sure you know the company, Plant Cell Technologies. They sell, ingredients for tissue culture.
Mm-hmm.
They were talking about synthetic seeds,
Mm-hmm.
and I saw synthetic seeds on your website. So please tell people about synthetic seeds, because I can't be the only one that doesn't know much about them.
Okay, it's very simple. Think about it this way. We can make a gel, right? That's what the agar is that we put in tissue culture, right? Normally, what we do is we make the gel. It's a liquid. We pour that gel into a container, and when it cools, it solidifies, and then it becomes the gel that you can tap, and it's a solid, right? Where synthetic seed says is like, why don't we just put the plant in the middle of that gel, and then we put the gel around it? So it's not a real seed. It's not new tissue that's made from two parents and the result of pollination. It's not a seed. You don't germinate it like a seed. You don't crack it like a seed, all that good stuff. It's a seed in that it's round. It looks like a seed. It's aesthetically pleasing. Most of the time in industry and in a lot of academic labs, they don't go for that aesthetic route. They just encapsulate it. Literally, they make the seed, the capsule, they put the node in the capsule, whatever shape it is, whatever shape it naturally made, because it's a chemical reaction. So when the two chemicals interact, it immediately makes that hardening of the shell. So how it works is, in traditional tissue culture, you put all your nutrients and hormones into the agar, and then the agar solidifies as it cools. In synthetic seed, you put all of your nutrients into the solution, and the solution will naturally solidify when it comes into contact with the second chemical. So it's a chemical reaction. It's not about cooling down. It happens at room temperature versus like tissue culture. When you pour the media, it has to still be warm. And yeah, they call it a synthetic seed because the plant isn't really actively growing. You can put it in the fridge. You can store it for a very long time. It has some similarities in a functional way as a seed, but like it's not going to have a taproot. It's not going to have any of that type of characteristics, and it looks immaculate in pictures. People love it.
Yeah,
very, very cool. Science fiction.
I mean, is this something that, for instance, out here, there's a lot of people that will grow seasonally and they take the winter off, like people that are not growing indoors.
Yeah.
Is this something that actually could be used for them?
No. So like,
from this, you still have to do the tissue culture process.
Yeah, exactly.
Is it more complicated than if you were to do tissue culture right out of the gate?
It's literally nodal. I mean, you're encapsulating a node, and then you're storing it. And when you break that storage, that dormancy, you're just doing nodal. What it really is for is like, let's say I wanted to get some genetics to Sasha. Even shipping stuff in tissue culture could be very temperamental to the temperature outside. I mean, because in tissue culture, the nutrients and hormones are still promoting growth. Growth under stressful conditions like the below freezing temperatures that it currently is means that you'll get messed up morphology or more commonly often death. Because synthetic seeds are about stasis, I can ship them somewhere that will take a very long time. They can get caught up in customs and then eventually make it to his doorstep. And then if he, can do nodal on them, he can germinate them. They'll have, the longer they stay, the lower germination rate there'll be, just like traditional seeds.
So this is a great way of transporting genetics to someone on the other end that can do nodal.
Yeah, this is essentially how all tissue culture organisms, I'm not even saying plants, this is basically how they're all sent. They're sent in culture. If they can survive in a petri dish, they'll survive in a petri dish. But if they can't, they'll usually be sent in some kind of suspension. The plant version of that suspension is a synthetic seed.
Wow.
It's super cool.
Yeah, everything you're doing is super cool. Something that I mentioned earlier before and wanted to make sure to get back to was, well, first of all, let me tell a story that someone else tells.
Hell yeah.
And this is Not So Dog. He tells a story about how he bought a bunch of plants that were tissue culture plants, and that, you know, it was thousands of plants at a giant farm in the Palm Desert or somewhere down in, you know, Southern California.
Lucky him.
And he said that he had a massive incidence of hermaphroditism. And he felt that it was because of the hormones that are used in tissue culture. And my buddy Cosmic, not because of that, but just because of lack of knowing how to, you know, mix the hormones or, you know, the fear of it, he just does nodal tissue culture with the MS, is that what it's called? And it works. Now, the disadvantage that I see is that the plants root and they kind of grow like a regular plant in there, whereas when you use the growth hormone, for instance, you get a lot of multiplication, so you get more shoots. But, you know, I'm trying to learn, and when I'm thinking about like what he's doing, I say, well, maybe this is less likely to cause a problem because he's not using these hormones.
Sure.
Even if he doesn't have the advantage of being able to get it to multiply as fast.
Yeah.
So, you know, what do you
I totally
think?
get where you're coming from. So, this is honestly more a broad question about hormones, their use, their function. How do we navigate it, right? Because we don't really use, you know, we're not plants. We don't use IBA. So, how much is the right amount of IBA versus how much is going to get you adverse effects, right? The thing we're always trying to not get any side effects, adverse stuff like that, right? So, when we started in Massachusetts, similar to Connecticut, non-agricultural state. That aforementioned plant, maybe I talked about like an hour and a half ago, Aronia, is grown primarily in Massachusetts. It's a tree. I never heard of it. It makes juice that makes cranberries taste sweet. So very unpalatable.
I have it here at home. It's growing outside of my house.
I mean,
Man.
it's a beautiful
Yeah.
tree.
It
But
is,
like in terms of like, you know,
Is it,
Yeah.
they sell pills of it?
yeah,
like-
it's very healthy for stuff like that. Yeah, like
Antioxidant.
I tried it like it was like a novelty thing where, you know, you eat it and then you try other foods and they taste completely different. And sour stuff will taste sweet.
Did it work?
It worked.
That's great.
It was
know-
crazy. I think it's called like Miracle something.
So I know a lot of people like that because it's a red fruit, so it's very high in antioxidants. And if you're someone who like really believes in oxidative stress, then it makes a lot of sense to like it. But it's not very common. So yeah, Massachusetts did not have a strong agricultural program. So when they legalized cannabis in 2016, what they did was they copied the USDA rules for organic horticulture blindly. 1-to-1. They didn't revise anything. They just copy pasted it. This is now their hemp program guidelines, right? So the USDA organics guidelines say you can do a lot of stuff. You can use salt fertilizers. You can grow in rock wool. You can do whatever, right? The only thing you can't do is use synthetic pesticides. It's the only thing that will make an organic grow not organic. Synthetic pesticides. Okay. One of my favorite things to talk about is that IBA is often synthetic. And auxins in high doses are herbicidal. So it is, by the USDA's definition and in the state of Massachusetts, IBA is a synthetic pesticide. You can't use it in tissue culture. You can't use it at all. They don't clone with Clonex. They don't clone with Formadin. They
Well,
don't.
maybe they do, and it's just hidden, right?
No, they don't. Because let me tell you,
You can test for it.
when when uh, when they were doing an audit in like 2018 or something like that, they found it in a facility, like a $4 million already licensed facility. They pulled their license. They didn't suspend it. They revoked it after they had gone like 3-4 years. And that was, so like, it was one of those, like, they led by example. I think most other facilities, especially because Massachusetts has essentially a $2 million entry, like dollar for entry.
I don't think you're supposed to use it in California either, and I think everyone uses it.
I think
I'd have to check.
It's all about how so some states, they, they legalize it accordingly. They say, if you use it as a plant growth regulator, it's not a pesticide. If you use it as a pesticide, it is a pesticide. Right. And they'll ban the usage and sometimes dosages. But some states will just, blindly ban it. That was the approach of Massachusetts. Which means when we started in Massachusetts, because it was legal in Connecticut wasn't, which is why I moved there and started in Massachusetts, we couldn't use hormones, and we didn't use hormones. And yeah, guess what? The plant doesn't need hormones. The plant has all of the genetic code and resources and tools to build whatever it initially had that we removed from it. It just needs building blocks, nutrients, literally just NPK, some micronutrients, some vitamins, and it'll do it. Hormones are where it gets complicated. And usually what I do in my classes when I talk about this stuff is I stop talking about plants, I start talking about people, because it's way easier for us to understand the difference between nutrients and hormones, right? So all of us can eat roughly the same diet in terms of grams of carbs, proteins, and fats, and total calories. If we all eat roughly the same diet, we'll all be roughly healthy. Few people, obviously, by percentage, have metabolic disorders. They need tailored diets. But generally, diet is very generalized. Hormones are different. I have a brother. That motherfucker, who is younger than me, is taller. When we were teenagers, if we both took HGH to be the same height, if we took the same amount, I might be taller and he might be too tall still, right? We need a dose amount. If we took too low of an amount, he might be taller and I might get cancer. Hormones are extreme signaling profile. I mean, think about guys who are now beyond that age of pubescence and are now like in their 30s or 40s. Like, you don't feel like you're 22 anymore, but if you get a dose of testosterone, like testosterone patch or HRT therapy, you'll have energy, your muscles will recover, you will have the appetite, your hair will start regrowing, your voice will get deeper, your memory will be better. All of those things are directed by your dose of testosterone, and all those things spike when you get a testosterone dose, and as you get further away from that injection or patch, you get back to your normal level of performance, right? Hormones are incredibly specific. They need a lot of diet. Same with, you can look all over Instagram. If you look at people who are doing tissue culture and their formulations aren't right, you'll get cancerous-looking calluses that are 3 times, 4 times, 8 times the size of the plant. That is clearly not directed growth of a regenerative plant. That is runaway growth, which in biology is the literal definition of cancer. It is growth that has bypassed a checkpoint to stop and just keeps growing. And as long as you give cancer food, sugar, which often, if you guys, you've made the media, is in these nutrients, they'll keep growing. So hormones can lead to all the difference in the world, right? They can make plants look phenotypically different after tissue culture when you transplant them. In fact, we just posted some pictures of super silver haze, the lost pheno, which is incredibly, I hate using this term, but I'm going to use it because I know it's industry standard, incredibly sativa dominant. I mean, it's not even hybridized. It's just straight up basically a sativa, or for phenotypic reasons, a narrow leaf plant, right? But if you look at the picture, its leaves are much broader because it just came out of tissue culture. And the hormones, cytokinins, have incredible impacts on leaf morphology and stuff like that in terms of keeping them green for longer, controlling something that's called programmable time death, which is what the leaves do when it's winter, right? They all fall off the trees. It's not coincidental. It's a mechanism called senescence. Cytokinins influence that. So out of tissue culture, the leaves of this super silver haze are much broader than you would expect. I know in like a month or two, as it gets farther and farther away from its tissue culture and hormonal experience, it will return to its natural hormonal balance and its natural levels. And the fan leaves will thin out. And this is something that we see across the board. It's actually why we posted the Jam It On 6, because it maintained extremely narrow leaves throughout tissue culture, which is extremely rare. It's probably the most narrow leaves that we've ever seen, which means it might be the most truest sativa we've ever worked with through TC, because it's very hard to pretend like hormones don't impact stuff, right? I mean, like, obviously. So that is like undeniable. To your buddies or to the story that you were talking about, right? Inheriting these plants and then them hermaphroditing out, it really depends on what hormones they use, because hormones have a shelf life. For example, Clonex comes in a dark bottle with 99.7% of it is not IBA. It's stabilizing ingredients, because IBA is very light sensitive, which is why it comes in a dark bottle, and very temperature sensitive. So you can't just willy-nilly have it in your grow room like you have a bottle of something else, because it'll eventually degrade, and now you've got a bottle of very expensive water. So you would expect that plants that leave tissue culture will 100% return to their true phenotype whenever those hormones get processed out. Some hormones, especially synthetic hormones, are way more stable, and if they're used, the phenotypic plasticity might last much longer than if other hormones were used. And that might have been what he was experiencing.
So, I mean, and I don't want to call it a mistake, but I guess what you're saying happened was, or saying is that these plants are much better used as mother plants than straight stock to immediately put in the field.
I mean, I have a very critical standpoint on hermaphroditism in cannabis. It's always the grower's fault. It's just that simple. Always the grower's fault. Because it's not a genetic trait. It's an environmental trait. So, I mean, and that's not to say, like, look, if you're growing 100 strains in one room, it's impossible to have all 100 strains have the optimized environments. You're going to pick some temperatures that some like, some humidities that some don't. And if you stress the wrong ones too far, that's what they'll do, because that's what a dying plant that can become male does. It chooses to
tries to save.
It tries to save its genetic. Like, DNA is selfish, and it's way easier to propagate your DNA. The whole point of living from a biological standpoint, I'm saying as someone who has no kids and no interest in them. So from a biological standpoint, like, that is the game plan. So yeah.
So what's your plan to survive, buddy?
Just enjoy my life with the wonderful people I have, you know?
Yeah.
And I know many wonderful family members that have kids, many wonderful friends that have kids, and I wish them all the best. I think knowing what you should do and knowing what you shouldn't do is a strength. So
I was just kidding.
I got you.
Yeah.
Your
But,
listeners get a
I also think you probably have a lot to offer. I feel like you'd be a better parent than a lot of the people out there that are parenting.
I hope to offer it to, again, my loved ones and friends, and to keep expanding on those people.
That's awesome.
But
I hope Sasha and I are part of that expansion program.
Of course. It's happening in real time, friends.
Yes, brother.
It's like literally the best part of this podcast is the friends that we're making. I forgot, he didn't finish answering the question about using hormones or not using hormones.
Yeah.
You know, because Cosmic does not use the hormones. He's getting them to grow.
Yeah.
Like,
you should use hormones. The reality is, like, the plant will grow. Just like in Massachusetts, if you clone in an aerocloner and you just put water in there that's pH correctly, and you just spritz it onto the base, eventually it'll callus, eventually it'll root, but those roots will be wimpier. They'll be thinner. They'll be more prone to developing the sheath, the little melanation, the little yellowing that happens, right? Hormones are growth promoters, at least the ones that we're talking about. You know, it's a very broad term. A hormone is literally just a messenger molecule that doesn't need a lot. It's the only thing that separates a hormone from like a nutrient. You need a lot of a nutrient, you need a good amount of sugar, you need very little adrenaline to get your heart pumping. So from that perspective, there are lots of hormones. Some synthetic that only exist in labs, some natural, and their wide appeal can be utilized in ways that have no detrimental effects. I mean, I know people who are doing tissue culture, they use hormones to keep their autoflower plants from autoflowering. So they're just photoperiod plants until they remove the hormones from their diet, and then they autoflower. And now it's, for them, for their propagative reasons, it's very, very beneficial. For someone who is inheriting someone else's work and doesn't necessarily know the history that these plants have gone through, yeah, it's certainly a risk to just put those plants directly into flower, but I think it would be a risk to put them into flower outside of tissue culture if you just inherit your buddy's garden for different reasons. Maybe less about hermaphroditism, although that could still happen if you don't validate the plants before you just flower a room with them, and more so like with russet mites or powdery mildews, this stuff that we talked about earlier.
I'm not sure, again, I think it's more what you said before, that it was probably something that the cultivators did, because this company doesn't sell plants that are in hormone. They sell, I'm not going to mention the nursery's name, but I'm sure you'll know who they are. They sell plants in flats that have tiny little, like the organic plugs, and they have foil, not foil, but like plastic wrap over the top.
The vented wrap. I know what you're talking about.
Right.
But the thing is, like again, hermaphroditism is always the grower's fault. It might not be the end grower's fault if you're inheriting a plant from someone else, but it is not a genetic trait, right? You can, and the easiest way to know.
Would you agree that there are some plants that tend to be more sensitive than others?
Sure. Yeah, prone. Yeah, that's fair. But okay, take your most feminine plant ever, literally the most feminine plant you've ever had that will never hermaphrodite under any circumstances. Spray that with silver thiosulfate. Spray it with colloidal silver. Colloidal silver and silver thiosulfate are not DNA. They're not impregnating. They're not genetically modifying the plant. They're shutting off certain regulatory aspects. The plant must still have the DNA to then produce pollen sacs if it's then going to produce pollen sacs. And of course it will, because again, it's not coincidental, it's mechanistic. So cannabis can at any time produce pollen sacs if the environmental conditions are
if it thinks
correct.
It has to survive.
Right. And it's your job, if you don't want pollen sacs on your plants, to not put those plants in those conditions. That might genuinely require you growing less susceptible plants to stress. I mean, it's really just like people. Some people handle stress very gracefully, and some people handle stress not as gracefully, right? But it's a personal thing. It's not a stress thing. Stress is universal. These plants have to be grown in an environment, so the cultivator has to decide what environment it is.
But you had mentioned about the cancer of the callus. Is that something that can be remedied, with time as well? Like if you have a plant that looks like that, or is that something that should just get? Excuse me?
Yeah. I mean, cancer is runaway growth. Just remove it from its source, and it'll stop. And then put it into a more appropriate source, and
it'll cut it off.
Yeah. The ability for this is really, really nuanced molecular biology, but this is called epigenetics. Plants' ability to differentiate, to go from a stem cell into any type of mature cell type, and de-differentiate, go back, is almost universal. They can go back and forth and from one to another and back into another and so on. This is why when you cut maybe a leaf close to where you're cloning, you'll see a root come out of the damaged petiole, right? Maybe sometimes you see, those cells change their identity. And sometimes if you see vines, they'll grow out of the ground where they're roots, they become shoots, and then they'll touch the ground again, and where they touch the ground, more shoots grow out. So they can go back and forth, whatever. Plants have extreme plasticity, and the hormones regulate that. So if you change the hormones, you just stop the cancer. It's radically different than obviously a mammalian perspective of cancer, where we're not plastic. So once something converts from one type of maturity to another, it's extremely hard for us to stop it and to get it to another type or to something else. Plants don't have that issue. So just remove it from your cancer-inducing media, change the nutrients around, the hormones around, you can put the same plant right back in there and see if the callus will behave more appropriately and if you'll get more normal morphology.
Well, we're going to have to let you go in a couple minutes.
One question I would have, man. Just this one, because this is really interesting. 500 cultivars you are having in Grateful Head project.
Yeah.
Right? How do you maintain? Is this like the same agar for all of them? I don't think so, right? I mean, because we are talking about like the load of hormones in particular cultivars. Some cultivars like this, some cultivars like you said, you know.
Yeah.
How you do that? How you maintain like 500 cuts in tissue culture with a program? Yeah.
What we initially did was, okay, you know what a Venn diagram is when you have the two overlapping rings? So it's like two circles where this is distinct, this is distinct, but the overlap shares, right? Okay. If you think of the Olympic rings as 5 sets of overlapping diagrams where they're all distinct but also have some similarity to their neighbor, that's how we started. We had 5 different medias, and what we did was we stopped presuming we would know what the plant wanted. We would blindly put it into the shotgun approach. We'd throw everything at the wall, and whatever stuck, that's where our next level of optimization would go from, right? We have been very fortunate to have worked with quite a number of cultivars at this point that we actually do have a universal media. So that's what we do. We just do one media for everything, just like the cultivators that I was so critical of growing all the same strains in one room and then potentially some hermaphroditing out. It is, some will like that one media more than others. None of them have shown in our experience, and there's always the next plant to prove me wrong. But thankfully, this media has worked exceptionally well. If ever comes a time where a plant doesn't like that media, we fortunately have this wide shotgun approach to fall back to, but as you can presume, for optimization's sake, it's incredibly hard to make 5 different teas for your plants or 5 different nutrient compositions, stuff like that. So we did focus a year and a half on optimizing to a single media, and we're very thankful to have done so.
Yeah, thank you, man, for the answer.
Yeah, that is something I was thinking about too. I'm glad that Sasha brought that up. I wanted to say that, as with most of our podcasts, we've spoken now for over 2 hours, and I know that we haven't scratched the surface. We like the long format here, so I hope we can get you to come back and continue the conversation. I also wanted to ask you about possibly joining. Wolf has another podcast on our network called The Way We Grow, and maybe just doing. Because that show is a lot shorter, it's like an hour, but basically maybe just giving a basic rundown of tissue culture, different ways people can do it.
Sure.
More for the, I don't want to say entry level, but just to inspire people, because I do believe that it's a valuable tool, like all the other tools that we use. Before we do go, I don't think we talked enough about the Grateful Head, how you became involved with them, what your position is with them, and we've had other guests like Kevin and Mark, so hopefully our audience is a little bit familiar that you guys are a repository for genetics, and you're preserving them, and you're selling Gen 0 clones, which are probably they're the cleanest clones you're going to get. They may not be sanitized, as we discussed earlier. But how'd you get involved with that project? And I'm sure you're very excited about it.
Yeah, absolutely. So I think most of the members maybe that you talked to were pulled in by a guy named Steve. And I was pretty unique in that they had a relationship with the lab already, and for whatever reason, there was a falling out in that relationship, and because they believed in the goal that they set out to do, they looked for another lab, and there's not many tissue culture labs. There are a number of labs doing tissue culture. They're most often vertically integrated into a grow facility, often into an MSO, multi-state operator. So being a third party and getting your genetics into their hands and allowing them to clean them up is often very hard. Not only like from potentially a trust situation, but really just from will they even do it? So Jason reached out, Skunk VA, reached out to me because we had the relationship with the class and basically asked if we wanted to be a part of it. And it was such an amazing opportunity. There was no way that we didn't want to be a part of it. Jumped right on it. It's a pretty amazing collection of guys. It's, like many startups, it's incredibly fast-paced. Things feel like they're changing all the time. Fortunately on my end, so I'm the Chief Scientific Officer, CSO, because I work with the plants. It's very, very easy. I just make sure that the plants look good. Everything, like you said, Gen Zero, so we go everything through Meristem, and then we sell those as clones to people. We've gotten incredible feedback. Everyone's been really, really kind and really receptive to the project. My heart goes out to all the C-suites outside of me who deal with people, find customers. That's always the toughest part of business, in my opinion. I'm always very thankful to just look at plants, enjoy being in rooms with plants, even now, and I hope to continue, with this project for as long as it manifests in this amazing way.
Well, that's pretty awesome. I think while you may not enjoy it as much, you're more of a people person than you give yourself credit for. So,
I'm more like a rambler, according to my family.
hey, you know what? I think that's a theme on our show. Everyone we get is a rambler. What I'll say is you speak a little bit slower and you allow us to, there's breaks for us to ask questions. I laughed that, we had Michael Wolf-Siegel and Kevin on the show. And those guys, they have so much inside that they want to say that literally you got to be like, you just listen to them. And, we loved it. I can't wait to have, Wolf is going to have his own show on our network. And Kevin's going to be invited anytime he's got, time to do it and has something to say. So, and the same goes for you. Like I said, I hope you'll come back and we can finish this story. And I definitely think it would be cool if you did, an episode of the Way We Grow podcast. More, I don't want to say for entry-level people. It could be for cultivators that have been growing for a long time, but really don't have the knowledge of what tissue culture is.
Sure.
And, I always tell our guests that, this isn't an infomercial. We don't do a lot of plugging, but I really believe in, what you're doing. And so I highly encourage people to check you out on, it's called the Cannomic, correct? How do you spell that? Your Instagram and your business name?
Kinomic.
Oh, Cannomic, right, Cannomic on Instagram. And you can go to his website and see what the services, the classes that they offer. And of course, check out the Grateful Head, for their genetic repository that is growing every day, and they're starting to get the clones out there.
Mm-hmm.
Anyway, thank you for joining us, Phil and Sasha. A warm hug from California.
I'm jealous.
Anyway, thanks a lot,
For
guys.
having me.